Lab Practical 1 Review Lab 3. In Lab 3, we talked about cell chemistry, the chemical composition of cells. This particular lab starts on page 35 of your lab manual. Some of the material on page 35 that you'll definitely want to be familiar with is the fact that some carbohydrates, all proteins, and no lipids are going to be polymers. These large organic molecules that we call polymers, or any of these macromolecules, are formed by dehydration reaction, where water is a product in that particular type of reaction. We're going to take our monomers, our small units, and join them together by a dehydration reaction to make a polymer. When we're breaking down a polymer, we're going to do just the opposite. We're going to have a hydrolysis reaction where water is a reactant and it is used to break those bonds that are holding that polymer together, and eventually we can break our polymer down into the individual monomers that we have there. One of the other things that we talked about before we actually started the experiment is found on page 36. It's talking about a control. What is a control? Whether we have a positive control or a negative control. Typically, a positive control is set up so that we definitely get a reaction. A negative control is set up so that we don't get a reaction. Often our negative controls are water. Instead of adding the substance that we're testing with, we will add water, and that is how we know if our reaction is taking place correctly or not. If our negative control reacts, then we know we probably have some kind of error in our lab experiment. When we're talking about a controlled experiment, typically we're talking about having this negative control present in our experiment. When we started working on our tests for the various chemical compounds, one of the things that we talked about were carbohydrates. Sugars, starch, glycogen, those are various types of carbohydrates that we talked about. We talked a lot about glucose and carbohydrates in general being a source of energy for the cell. Here we see just a quick example of if we move the reaction one way with one enzyme or one set of enzymes, we can have a dehydration reaction where we're putting larger molecules together. Water is a product in that reaction. Or we could run the reaction the opposite direction, taking a larger molecule like a polymer and breaking it down by hydrolysis, where water is a reactant, and we're breaking it into the monomer units eventually. We did two different types of tests for carbohydrates. One of those tests is found on page 37, and we tested for starch. To test for the presence of starch, we used iodine. Iodine starts off that yellowy brown color, and if we have a positive reaction, if starch is present with the iodine, then the solution is going to turn a blue-black color. So if starch is present in our solution, then the iodine changes to basically a black color, as table 3.1 indicates. We also tested for sugars. When we test for sugars, we use Benedict's reagent. Benedict's is a bright blue color. This particular reaction has to be heated before the reaction will take place. The reaction that we see gives us a little bit of an idea of how much sugar we have. Not only do we know if sugar is present or not, but we know an approximate, well, at least a relative amount of sugar. If we have a very low amount of sugar present, then our Benedict's goes from blue to a greenish color. If we have a high or very high amount of glucose or other sugar present, then our solution is going to turn our orange or orangey red color. So we have a little bit of a relative amount. We know a little bit of sugar, a lot of sugar present when we're testing with Benedict's. Moving on to our next type of chemical structure, a macromolecule, I should say, is protein. Protein is made out of monomers. All proteins are going to be polymers made out of the monomer amino acids, all hooked together with special types of bonds referred to as peptide bonds. That's where we come up with the term polypeptide, which would be a large chain of amino acids. Some polypeptides function and we refer to them as proteins if they actually function. If it's just perhaps even a random distribution of amino acids all put together, then we have a polypeptide with that. Some examples of proteins or polypeptides, enzymes. Most of the enzymes we talk about in this class are protein. Albumin is a transport protein in egg white, and we did a test with that. When we tested for protein, we used the Biuret reagent. Biuret reagent is blue. It's not the same shade of blue as Benedict's. It's more of a blue-gray color. The Biuret solution will turn purple if we have protein present, or it will turn a pink color if we have peptides present. So if we have a functional protein, then the Benedict's is a little more specific and turns purple. If we have a non-functioning protein for whatever reason, the Benedict still will react with the peptide bonds and turn that pinkish color. We tested for fat. We did not use Sudan-4. Sometimes I use Sudan-4, but we did not use that. We just did a very simple test with brown paper, and if the brown paper stayed looked like it stayed wet, then we said the solution that we were testing had oil in it, and if the brown paper dried, then we did not have oil present or any other type of lipid present in the solution that we were testing. One of the things we did talk about with lipids is the fact that lipids will not dissolve in water. So if we want to get our lipids to dissolve in water, often we have to add an emulsifier. We have to have an emulsification. It's not going to break the lipid down and break it into its smaller units. Technically, they're not monomer units in lipids, since lipids do not have a repeating pattern. But they are made out of different smaller units depending on what kind of lipid we have. What the emulsifier will do is take a large chunk of lipid and break it into smaller chunks of that same lipid. It's not going to break it down into the components, but it will break it into smaller chunks. The example of an emulsifier that we used in Lab 3 was bile salts. Bile salts are produced in the human liver. They are stored in the gallbladder, released into the small intestine, and that allows the small intestine to mix the bile salts with any kind of fat that you're trying to digest, and it allows that fat to dissolve better, so then enzymes can come and kick in and resolve that. We did have a little homework on page 47. You might want to look over some of those questions. It's always good to go back, not only to review the material, review the quizzes, but also review the questions that you had at the end of the chapter. Sometimes they show up on the quizzes, sometimes they will show up on the lab practical as well. Especially the ones that people tended to miss the most. I might ask those again just to see who was really paying attention in lab, and then to see if you're paying attention to any feedback that you received in the lab. All right, so that's Lab 3.